recap oxidative phosphorylation. Now, previous lesson we were out of class, right? Yeah. I don't know why the previous lesson felt like a long time ago. How's your lab, is it? Okay, lab. Okay. But I can't remember what I did with you all. Not sure what you all remember. Okay, we were doing skates last round, right? We were trying to show each other how the last step of cellular respiration works. Okay, we didn't have time for me to show you this video to sum up the entire process. Okay, but I'm going to show this video now to sum up the entire process. Let's just take note of a few things. Again, number one, this chapter, we want to form the length of interactions. We want to look at where it takes place, what changes, and what is needed. Okay, with that lens, we're going to watch a video to sum up the last step of cellular respiration. There's also a lot of movement taking place. Okay, lots of hydrogen ions moving here and there. So I'd like you to take note of the movement, the changes that take place. If you want to, you can stand by your notes, go to the page of oxidative phosphorylation, and you should be able to take down any additional notes that you find interesting or new to you. So if you go to your notes, go under energetics, go to cellular respiration. And you scroll down to this part over here. Okay? The video I'm about to show you shows this part in action, moving. Okay? I suggest that when you revise this chapter, this part of the notes, watch a video. It's much faster. Don't try to read and understand. It takes ages. Okay? Not that reading is hard, it's just that some things are better represented as a video. So watch the video instead. And then here is the video of the process.
Most of the energy harvested from organic molecules during glycolysis and the Krebs cycle is stored in NADH. NADH and FADH2 molecules give up their high-energy electrons in the third phase of cellular respiration, electron transport and ATP synthase, where most of the cell's ATP fuel is produced. The electron transport chain is an array of molecules, mostly proteins, built into the inner membrane of the mitochondrion.
Okay, so pause here. There's one thing you want to take note, is that these purple things, they're all proteins. They're all protein in nature. What does this mean? It means that we can apply the concept of proteins. I could randomly extract one protein out and ask you, what level of structure do we see here? Or I could say that I heat up the mitochondria to very high temperatures. What would the effect be on the protein? Or if I throw in acid, what would the effect be on the protein? Okay, so just remember, they are all proteins.
ADH gives up its high-energy electrons to the first complex in the electron transport chain. The electrons move from one member of the chain to the next, giving up their energy as they are pulled from NADH toward oxygen. The energy given up by the flow of electrons is used to pump hydrogen ions from the mitochondrial matrix into the intermembrane space. Oxygen captures the electrons in the very last step in electron transport. The last complex adds a pair of electrons to an oxygen atom and two hydrogen ions, forming water. The electron transport chain has used the energy of moving electrons to pump hydrogen ions into the intermembrane space. This buildup of hydrogen ions, like water behind a dam, stores the potential energy that was originally in the bonds of glucose molecules. The backed up hydrogen ions give up their energy when they diffuse through a special protein complex in the membrane called ATP synthase. As hydrogen ions flow down their concentration gradient, ATP synthase captures their energy to make ATP.
Okay, a quick one.
Under normal conditions, almost all the ATP produced in the process of cellular respiration is manufactured by the energy that electron transport provides to ATP synthase. About 34 ATP Most
Okay, a quick one. As we look at this particular interaction, where does it take place? How will you describe where it takes place? Okay, a bit tough here, right? It's like it involves the intermembrane space, involves the matrix. Yeah? So we could say that it takes place across the inner membrane, between the intermembrane space and the mitochondrial matrix. That's the where. Who is involved? Okay, so what is involved at the very start of this electron transport chain? Okay, NADH. If you are unsure, just whack NADH because it's involved in a lot of places. Sorry, NAD plus is what's involved in all the steps, but we've been collecting NADH for this very final step. Okay, so what's needed? NADH is needed. FADH2 is also needed. They are all electron carriers. They've been storing the electrons for this very final step. Okay? Other things that are needed, oxygen. Okay, oxygen is also needed. So if you now you know to a deeper level, when you breathe in oxygen, it is needed just for one step, oxidative phosphorylation. It just acts as that last acceptor of electrons. We're going to introduce some vocabulary to you that if it's not in your textbook, you want to take note, obviously in your textbook you want to highlight it. Okay, number one, we refer to oxygen as a final electron acceptor. Make sense? Not too complicated. It's just a final thing that accepts all the electrons. Yeah? And if we look at the overall equation for cellular respiration, it's nothing more than glucose plus oxygen, and you get CO2 plus H2O. Glucose is doing nothing more than transferring its electrons to oxygen. Except we don't do it in one shot. We had a lot of intermediates that help to transfer this electron to oxygen. As a result, oxygen gets converted to H2O, and glucose gets converted to CO2. This is the overall reaction. And what we learned in primary school and lower GH. But for us, we've gone in a little bit deeper. We see that there are many intermediate steps that led to this process. Okay, that's why we refer to oxygen after all as the final electron acceptor. That's one of the vocab. Okay, so what is needed? If you want to look at what is produced, what is produced? Okay, just a quick look through of the reaction. What is produced at the end of this whole chain? Okay, H plus. Is H plus produced per se? Sorry? ATP is produced. ATP is produced. Water is one of the products also. Is CO2 produced in oxidative phosphorylation? Sorry? Okay, if you want to ask yourself, when does CO2 produce? It produces in Krebs cycle, it's produced in a link reaction. What else is produced? Another way to look at this, NAD plus as well as FAD. In other words, what is produced, you want to think of it is what is the change? Right? These are related to each other. Okay, the oxygen becomes water. There is a change. Okay. So once again, for this chapter, it's just about interactions. We don't need you to memorize every single step. It's just about understanding what goes in, what comes out. Yeah. Eat something combination. Probably not the best analogy.
ATP.
the electrons to move from protein to protein? Well, because we want to release energy to transfer, to pump the hydrogen ions into the intermembrane space. So you read through your notes. Some of the vocab you can use to describe the process is pump. Okay, this is one vocab you can use. Pump. Okay, as a verb, we pump hydrogen ions from matrix into the membrane space. So this is one of the vocab you can use. Pump. The more we pump, the more hydrogen ions will accumulate in the intermembrane space. Right? In a sense, we set up a difference in concentration. We refer to that, the next vocab to use is photon gradient. It's not in your notes. Okay? Photon gradient is just a short form way of saying difference in concentration gradient of the hydrogen ions. Instead, I can just say photon gradient.
Difference in concentration of what hydrogen?
Yeah. Okay, let me give you an example to use in a sentence. Okay. So you listen up. As electrons are transferred down the electron transport chain, energy is released to pump the hydrogen ions from the matrix to the intermembrane space. As a result, we set up a photon gradient. Okay, so that's how I will use it in a sentence. We set up a photon gradient. Okay, want to encourage you in biology, it took me many years to figure out that really don't memorize the sentences, waste memory space. You really want to memorize things, memorize the vocab. Then you use it in a sentence. Right? Just like you go for Chinese exam, right? It's not like you memorize sentences. Okay, I admit at one point I memorized sentences. When I go for my mother tongue exam. But usually we'll remember the vocabulary we use, right? Then we string it together in a sentence. Same for our biology. Yeah, one may be wondering, what's the point of accumulating so much photons up there? Right? What's the point? Well, because ATP synthase is very unique. This enzyme only works when photons flow through it. Then in our minds, right, we are trying to visualize, how can this happen? How come when something flows through, it suddenly works? Yeah, I'm going to show you how it works. You know, sometimes we running river, sometimes people put a wheel there, and then you catch the water and you move along. The ATP synthase works kind of like that. Let's watch.
Synthase is a molecular machine that works like a turbine to convert the energy stored in a proton gradient into chemical energy.
See, as the photons flow through it, it gets the whole ATP synthase moving. As this part moves, it changes the shape of the bottom part also.
The flow of protons down their electrochemical gradient drives a rotor that lies in the membrane. It is thought that protons flow through an entry open to one side of the membrane and bind to rotor subunits. Only protonated subunits can then rotate into the membrane, away from the static channel assembly. Once the protonated subunits have completed an almost full circle and have returned to the static subunits, an exit channel allows them to leave to the other side of the membrane. In this way, the energy stored in the proton gradient is converted into mechanical rotational energy. The rotational energy is transmitted via a shaft attached to the rotor that penetrates deep into the center of the characteristic lollipop head, the F1 ATPase, which catalyzes the formation of ATP. The F1 ATPase portion of ATP synthase has been crystallized.
Can you see all the alpha helixes? Yeah? And this is after a protein. Classical music. When I see this, I was very amazed that this is happening on a microscopic level inside a mitochondria on the inner membrane. It is crazy. I had the privilege once of meeting the person who discovered this entire process. I asked him, well, how long do you spend discovering all this? He started working when he was in his late 20s. Now he's 70 plus. His whole life he dedicated to discover how this process works. Every few years he'll discover one new part how it works. One new part how it works. He discovered the first part how it actually turns. Then subsequently he realized it's a shaft. Then subsequently he realized down there also changes. He spent his whole life, now his hair all grey already. He won a Nobel Prize for this, you can expect. Because at the end of the day, this really helps us understand how our body synthesizes ATP. I'm actually taking a photo of him. Oh, but I never bring a photo today. I'm so excited. Short of asking him, can you show me your Nobel medal? Okay, so that's how the entire process of cellular respiration works. We see that therefore, actually, you know what's interesting? At the end of oxidative phosphorylation, we produce the very same things that we need to kickstart the whole process again. Right? Glycolysis, link reaction, Krebs cycle all requires this, right? NAD plus. So we come full circle. So if you think again, you know what the problem will be if you stop breathing? If you stop breathing, that whole chain has no one to pass the electrons to. If there's no one to pass the electrons to, in the final step, no more protons will be pumped. No more protons will be pumped, no more ATP synthase will be produced. If there's no electrons, nowhere for the electrons to go, the NADH will not give up their electrons anymore. We will not what we say, regenerate NAD plus anymore. Then you will slowly shut down as a system. Okay? So the beauty about this whole cellular respiration process is that we produce at the end the very things we need at the start. And this cycle continues and continues and continues to keep us going. But the moment you stop breathing, or you don't have enough oxygen in your body, this whole process will start to grind to an end. Which is our which is one thing we're going to talk about today, anaerobic respiration. The idea that Na means, wait, what's Na mean? Na means I think don't have. Aerobic means if you don't breathe. So anaerobic respiration. What happens if you, okay, it's not exactly they don't breathe. Did I ask you all to do an SLS, the last part? No? Okay, very good. Can you raise your hand now? Okay, everyone raise your hand. Let's just do this. Really as fast as you can. And just do it for as long as possible. Let's see who can last as the longest. Okay, I'm starting to slow down. Some of you can still go pretty fast. Okay, even if I tell my arm to go faster, it just won't go faster. Like, go fast, it just won't go faster anymore. Do you feel your arm cramping up? Yeah? Do you know what is accumulating in your arm right now? Acid. You know what the acid is called? Okay, you know, my arm can barely move already. It's seriously cramping up. Okay. I don't think I can even do what you start. Hey, shucks, I'm right-handed. Okay. The thing you're accumulating right there in your arm that starts to hurt a lot, that's called lactic acid. To your last page of your notes, over here. Okay? We're going to talk a little bit about how our body responds to a situation when we don't have enough oxygen. You just simulated an environment where there's not enough oxygen. What do I mean by that? You are clenching so fast that your arm needs a lot of energy, right? But you know what's the issue? You were not breathing fast enough to keep up and supply enough oxygen there. So, oh, okay, actually, someone got a point there lah. Oh, your arms will cramp during exam. Okay, I remember I always cramp up, right, for my geography or history exam. Right, my whole hand will cramp up. Then after I'll take a break, then I'll do this once in a while. then write some more, then after that, then come back. Yeah, that's how I felt. Okay, so breathe more. Okay, I won't say breathe more you can. But then other than breathing more, you need to get the blood pumping to the area. So the reason why I ask you to raise your hand is because it's much harder for your blood to pump upwards. So that your arm are really deprived of oxygen. And that's when you start to feel the effects of anaerobic respiration. So, yeah?
If I lie upside down, then I can rise up.
Okay. You go ahead during exam and do that. I'd like to see you do that. So our body needs to respond and cope with this situation, right? Not enough oxygen. If there's not enough oxygen, we will stop regenerating NAD flux, right? Okay, so how does our body cope with that? Copes with it with a very interesting idea. In glycolysis, right after pyruvate is produced, it does something interesting. Our body then converts pyruvate to lactic acid. It converts pyruvate to lactic acid. I know what happens when it converts it to lactic acid. It regenerates NAD plus. Okay? NAD plus is regenerated when you convert pyruvate to lactic acid. And this allows glycolysis to continue on, and continue on, and continue on, to produce a little bit of ATP. Just a little bit of ATP. This NAD plus that's regenerated is not enough to power your Krebs cycle or your link reaction, but it's just enough to power the first step of cellular respiration, just the glycolysis step. It is enough to keep your arm moving a little bit more. It's not a long-term solution. So if I ask you to hold your breath, eventually you'll meet your demise. Okay? That's it. And regenerating NAD+. So you can use in a sentence, pyruvate is converted to lactic acid, regenerating NAD+. Okay? To allow glycolysis to continue on. And that gives your body a little bit more energy to last a little bit longer. Until you catch your next breath. Or until your heart beats faster. Yes, Rachel got a question. Okay. We call this process lactate fermentation. The process of producing lactic acid to regenerate NAD+. It's called lactate fermentation. You can highlight that for yourself. We aren't the only animals or organisms that can carry out lactate fermentation. A lot of animals can too. However, not all organisms will convert their pyruvate to lactic acid. Sorry, you have a question I want to clarify first? You have? Yeah? If it's oxygen, supply that. Okay, if there is oxygen, would it? Oh, yeah. Correct, okay. So maybe I wasn't clear. This process kickstart only when there's a lack of oxygen. Okay? Only when there's a lack of oxygen or zero oxygen, then we will kickstart this process. Rather than let glycolysis go on to the next step, to link reaction, to Krebs cycle, we will instead, run glycolysis through this alternative reaction. To keep your body going, because the body knows that there's no point in running it to the last step Because there's no oxygen. Okay, good question. Turns out other organisms can also carry out a similar process when they are deprived of oxygen, except that they don't convert pyruvate to lactic acid, they convert it to alcohol. So you scroll up. This is another process, another alternative, can convert it to alcohol. But other than converting it to alcohol, it will also release some bubbles of CO2. Do you know when we can actually make use of this process to produce something we eat? Not bread. No, alcohol plus bubbling gas. Yeah, like wine. So I don't know what the first person was thinking, but I last heard, right, a long time ago when you make wine, right, is they take grapes and then you step the grape, right? Yeah. You know what's in your feet? Lots of bacteria. Right? These bacteria, when you deprive them of oxygen, you know, okay, not bacteria, there's a lot of yeast, fungus. When you deprive them of oxygen, you know what will happen? They will kickstart their version of what we do. Except that the byproduct is alcohol and CO2. So honestly, I don't know what the first person was thinking. After they stepped ready, then they thought to themselves, you know what, I'm going to keep this and not drink it immediately. Then 1 month later, they drink and like, wow, it's really good. Yeah, so wine. What else? Anything that's alcoholic tends to be produced this way. Your wine, your beer, your vodka, I don't know vodka. What other alcohol? Soju, what? Sorry? Whiskey. You know what changes? What changes is what is being fed to the yeast. Yeah? In wine, you feed the grapes. In beer, you feed malt. We change what is being fed. But what's the constant is that we suffocate them. Deprive them of oxygen until they produce alcohol. Yeah? I once left grapes in my... Wait, I told you this story before. No, I once left grapes in my fridge for so long, it did not mold. But after a few months, right, I just forgot about it. I left it at the bottom shelf. But it did not mold. Yeah, I just took out. It's still okay, but I can't remember where I bought it. Put it in my mouth. But it was fizzy. It was slightly alcoholic. I don't encourage you to do that. I'm just saying that it is possible. Yeah. Unfortunately, I'm allergic to alcohol. So the more I drink, right, the more I'll flush, and then the more my airways start to tighten, and then I'll start to wheeze and I cannot breathe. So, hey, what you say is that? Yeah, it's a good and bad problem. So there are days when I was in like JC it's like so havoc, right? People want to go and club, right? They're like, hey, you want to go or not? Then I say, cannot. I will like, I may die. Then, how people go? No, you all shouldn't ask, sorry, yeah, I'm being recorded, so you all shouldn't ask. I'm just saying that there were naughty kids, but I very good one. But I also don't know how to reject them, so I tell them that if I drink alcohol, I will die. It helps. So if you don't want to go to anything, just tell them you're allergic to alcohol. Okay, but then there was one time they asked me, hey, just go there and without alcohol. Then I'm like, I think it's a weird feeling to do it without alcohol. Okay, so we have come to the end of the second last chapter, cellular respiration. Give me a while, I'm going to go to the next class to collect some items from this thing. Right, and then we're going to do a demo to kickstart the next chapter. I'll give you some time to touch up your notes. Photosynthesis. Okay, download your notes. And we're just going to do a quick introduction to this chapter. Okay, so this is the last chapter for this term, photosynthesis. If you look at this chemical reaction in your notes, notice that it looks very similar, right? The products and reactants look awfully similar to cellular respiration. It's just that we kind of invert the process, right? Yeah, notice, right? We use CO2, we use water, we produce sugar and oxygen. Wasn't cellular respiration just like at the reverse? Yeah? So, photosynthesis is this magical process where we can produce sugars, right, at the end of the day. And if cellular respiration was all about oxidizing glucose, this is about reducing carbon dioxide to create sugar. It's really just a redox reaction at the core of it all. And therefore, you will find many similar processes compared to cellular respiration. Right? So for this last chapter, we can go a little bit faster because you will find an electron transport chain also. Yeah? And that's why we spent a little bit more time in the previous chapter on the electron transport chain. But today, to truly appreciate how plants are able to make use of light energy to, at the end of the day, produce sugars, must appreciate the pigment that is able to grab hold of this energy. And that pigment is, sorry, it was very anticlimactic, just for replying. And that pigment is, okay, it's chlorophyll. The greenness you see in every single plant, chlorophyll. Okay, chlorophyll is a pigment that allows the plant to trap light energy. If you want to know what chlorophyll looks like chemically, you scroll down to this particular page over here. It's a very complicated chemical. You know what's the interesting thing? The hemoglobin pigment, hemoglobin pigment, there's a pigment in your blood that makes your blood red. Hey, that's hemoglobin, right? Wait. Yeah, I'm going to Google hemoglobin. Hemoglobin. I recall searching this up before. I'm wrong. Hemoglobin and chlorophyll looks, I found very interesting. Structurally, right, it looks almost similar. This is what I found interesting. Yeah, sorry, not related per se, but I just find it very interesting that it looks quite similar. Except our body, we have iron in the middle, and for plants, right, what they have in the middle over here is magnesium. Yeah? So, actually, on the fundamental level, chemical level, we are in some ways similar to plants. Yes? You want to create a human plant, right? I've thought about it before. Okay, but I suspect, right, again, it doesn't work because we just need too much energy. So, just photosynthesizing, right, it's not enough. If we rely just on photosynthesis, right, you see people running on a track, right, after they collapse already. You set yeah. And also, you think about it, I think the surface area is just not enough for you to produce enough energy. Yeah, but plants are the leaves are very flat and they have lots of leaves. If you really want to photosynthesize, you need a lot of arms. Or maybe you should put in a pen. Maybe just have a whole bush of hair and you can start run around with. Okay. So chlorophyll. Do you know where chlorophyll is found? Okay, yes, leaves. Okay, there's an organelle where the chlorophyll is found. Chloroplast. Okay, so let's look at the chloroplast today. Let's try and make sense of where you can find chlorophyll. You can find chlorophyll inside this organelle called chloroplast. A chloroplast is, in a sense, similar to mitochondria. It has an outer membrane. It also has an inner membrane. But the inner membrane is not highly folded like that of the mitochondria. Yeah? Last but not least, the chloroplast also has one more layer of membranes. Yeah. Hey, you learned this before? Last year? Oh, okay, very good. Okay, so this last layer of membranes are all folded upon each other to create small little like M&M shapes. Okay, when I draw these circles, understand that they are like really like M&Ms. They are envelope to form like a small little bubbles. Okay? We refer to each one of them as thylakoid. The membrane that makes up the thylakoid is called the thylakoid membrane. I say again, the membrane that makes up the thylakoid is called the thylakoid membrane. So, where exactly is chlorophyll found? Yeah, I'm going to pinpoint it in red. You find chlorophyll on the thylakoid membrane. Okay, so the M&Ms are the real important fellas. Okay, they're the ones that have the chlorophyll found there. It's on the membrane. Okay, if we have time, I will show you where exactly it's stored. But today, we just want to talk about chlorophyll. I want to understand how we make use of this energy that comes from the sky. Nothing inside, is it? Okay, it's almost like a mitochondrial matrix. It's just there are a lot of reactants that's required. Yeah. A lot of hydrogen ions also, because there's setting up photon gradients. Yes? Okay, you see the word random on this particular page over here? Random is nothing more than a stack of thylakoids. Okay? We rarely use this particular vocabulary because when we describe photosynthesis, we will zoom in to one of the thylakoids. Every thylakoid lots of photosynthesis particles. Alright, to understand how chlorophyll pigments work, I'd like you to go to this particular page. The idea that electrons can be excited. This is so exciting. Okay. The electrons inside chemicals can be excited. And some electrons, when excited, okay, so this is the energy level. Energy level. Okay. And this is my electron. There are some chemicals that have this very special property. We can excite the electrons when we supply it with light. Okay, so this is light, energy. But when we supply the electron with energy, it gets excited. Okay? This arrow being drawn up represents it being excited. After being excited, right, there's only so far high you can jump before you have to come back down. After it gets excited, it will return back to its non-excited state. Okay? But that's not the interesting bit. The interesting bit is, when it goes back down to its non-excited state, it will release the energy that it came from the light. And it can release it in the form of heat or release it in the form of light again. Yeah? So literally, if I'm an electron, light shines on me, I'm like, woohoo. Then as I'm coming back down, right, I glow. And I don't glow, I release the light energy that I absorb. Okay? How is this useful? Okay. We'll see how this all comes together in tomorrow's lesson. But today I just want to show you what this looks like. If we supply electrons energy, let it get excited, let it go back to its normal state. I want to see this part over here. Okay, I'd like to get a volunteer for this demonstration. And I need to switch off the lights. Oh, that was a fan. Any volunteers? Don't worry, you won't hurt, I think. Oh, but it's matches glass. Okay, here I have very super powered light. It's UV light. You cannot quite see anything coming out of this, right? Yeah? But it's very high powered. You know, UV light is very powerful. In fact, it can cause skin cancer. Every time I shine it on my hand, right, I go home, right, I feel itchiness. Okay, it's very powerful. I would like here I brought a can of tonic water. Anyone drinks this? Tonic water is essentially like bubbling water. Yeah. Okay. In tonic water, it contains this thing called quinine. And I found out online that quinine contains electrons that can be excited if you were to supply with a lot of energy, light energy. Okay? I want to show you what happens when we supply quinine with light energy. May I get a volunteer? Hey, are you all pointing each other or both of you want to do it? Okay, yours is loser, come on. Hey, winner, come on. Yes, paper. So, alright. Okay, you see, thank you. Give a round of applause for you see. Okay, this is what you do. Thanks, you see, we'll drink this and then I'll shine the UV light on her. No, of course not. That's not going to work. Huh? Okay, so you see step number 1. Can you, like open the can? Let's say open. Okay, so you see inside there's a lot of quinine chemicals. Contains a lot of electrons. What I'm going to do now is that I'm going to shine light into this particular glass. Okay. So this is what we'll do. This UV light. I'm going to place it over here. You see it's going to pour it into this jar over here. Right? As she pours it in, we see what happens. Ready? 1, 2, 3. Okay, stop. Isn't it amazing? Not really, Wait, let me pour this a bit. Okay, now you see, I'd like you to pour it in as I supply this quinine with high energy light. Ready? Go. Beautiful, right? Okay, thank you. And if I were to just take it out of this high energy light, yeah? So what's happening here is this. The moment I supply the electrons in this quinine with the high energy light, the electrons get excited. At the same time of being excited, it will become unexcited. That's only so far it can jump, right? But when it goes back down, it releases the energy back in the form of light. You cannot quite see the light that I'm using, that I'm shining on my torch, but you can see the light that the electrons emit. Okay? Thank you. You can keep that can. You want to drink? You can give it to Sarah. Now, I've extracted something else today. Here, I have a whole jar of chlorophyll. Okay? Because chlorophyll also contains excitable electrons. You know what that means? If the electrons are excitable in the chlorophyll, that means I can also supply with high energy light. Okay, so what will happen now when I supply with high energy light? Ready? Want to make a prediction? Okay, let's see what happens last. Okay, lights on. Can you see? It doesn't quite glow, but it releases a very low red colored light. Can you see that? Almost like blood now. Yeah? Right? Almost like blood. But if I shine it light, can you see it's green? It's very much green. So the electrons in chlorophyll contain the same property. They can be excited, and when excited, they will be excited, and then when they come back down to their non-excited state, they will release light energy. But does this apply if I will shine this onto the plants outside? Right? Okay, let's do that. Okay, here I have a leaf, right? There's a lot of chlorophyll in this leaf. So if I were to shine light onto it, what will happen? It should glow, right? Okay, so high energy light, Nothing much is happening, right? Yeah? So we have a conundrum here. Why is it when I extract out the chlorophyll, put it in a jar, and I shine light onto it, the electrons get excited, goes back to ground state, and it can release energy in the form of light? Because the energy is used. Right? The reason why our plants don't glow red is because the energy after the electrons get excited, and when it's going back down to its ground state, it doesn't let the energy get released in terms of heat or light. You don't feel the plant heating up. Instead, the energy that's released when electrons go back down to its unexcited state is used for a meaningful purpose. And that meaningful purpose is eventually produce sugars. Okay? So, the reason why I show you this demonstration today is just to give you an understanding of how plants make use of this energy. It's able to make use of it because its electrons are excitable. When it gets excited, go back to ground state, the energy that it releases, it will use it. It doesn't let it go to waste. Okay? So, I don't have much time to go into the deeper into it, but we'll continue tomorrow to understand where the energy is used. Okay? We need to understand where that energy is used. If you want to appreciate tomorrow's lesson better, may I invite you to read this particular page of the textbook. Okay? Aziz asks, if I shine light long enough on the disc, will it taste sweeter? Actually, in theory, it should, right? Yeah? More and more sugars being produced. Yeah. Okay? In tomorrow's lesson, this is what we're going to learn. We're going to learn how the plants are able to capture the released energy. Right? And then we will see how it funnels into everything else tomorrow. Okay, thank you for today. Okay, I want homework which is cellular respiration RE. Okay, by this Saturday or Sunday. Okay, thank you class.